Good day, everyone. I am Francis Opoku. I present to you the contribution of cover crops and soil test phosphorus levels to phosphorus loss in rainfall runoff. The green substance you see here in this cup, I'm sorry, is not a fruit juice, so don't test for it. This is algae in water, and this is caused by the excessive loading or enrichment of nutrients in surface waters, most especially phosphorus, that the algae feed on to grow. When there's the bloom of algae, there's the problem of reduced water quality, therefore fish by limiting oxygen conditions. There's also human health risk attached, including so many other negative effects. Lake Erie, which is embedded in southwestern Ontario, where my research is done, is the warmest and the shallowest of all the 5 Great Lakes. And because of these conditions, it is the one that is suffering the severity of the problem of algae growth. Agriculture has been identified as the major long-point source of phosphorus. If there is a source, there should be a transport mechanism. And the transport mechanism found include soil erosion, surface runoff, and tile drainage. Earlier on, efforts at mitigating phosphorus loss were focused on soil erosion control, where cover crops were applied as one of the best management practices in reducing particulate phosphorus losses, that is, the phosphorus attached to particles of soil, because the cover crops serve as a barrier to filter out this phosphorus. And this was very effective in the fall and also in the spring season. But then it was realized that the actual transport medium, which was the problem, later on was found to be runoff. And also, it's not just any phosphorus, but what the form of phosphorus called dissolved reactive phosphorus is the one that algae loves to feed on. And therefore, new targets were set to look at reducing both total phosphorus and dissolved reactive phosphorus by 40%. And this was in reference to the amount in 2008, which was 10,000 metric tons. This is to be achieved by 2025. We are getting there. Cover crops, although, can reduce the amount of particulate phosphorus. However, they have been found to also contribute to dissolved reactive phosphorus. In what sense? In winter, when cover crops are left on the soil to provide cover, there is breaking of the cells of the soil by the freezing and thawing temperatures. So when the cells of the plants, not soil, when the cells of the plants are broken, the nutrients in them are released. But whether they join runoff into surface waters is still debatable. Works in southwestern Ontario, Coba and Lozé, with different cover crops, soil test phosphorus levels, they have found that cover crops may have little to no significance because the amount that is lost in the soil itself, the soil water soluble P, compared to what is lost in runoff when even there is cover crops on the soil, is very, very low. However, Elliott and Beckman, in different geographical areas with different freezing and thawing conditions, different soil test phosphorus levels, when you compare, for instance, annual white gas to bare soil here, there is a big difference, and therefore soils that have annual white gas on contribute more dissolved reactive P in runoff than bare soil. Because of this, we set out this objective to look at the effect of cover crops on the loss of phosphorus in snowmelt and rainfall runoff from agricultural lands under southwestern Ontario conditions, where we are looking at different, there is frequent freeze-thaw cycles, but very little to no snowpack. Also, different range of soil test phosphorus levels. Soil test phosphorus is the measure here because it is the phosphorus in soil which is bioavailable and therefore bacteria love to feed on. Also, there is different cover crops that we are testing. Watford sandy soil were packed at a depth of 8 centimeters in these plastic pails, and a slit was cut here to allow the movement of water once we put the snow or when we put the rain on the soil. Three cover crops, which were red clover, oat, and annual ryegrass, and a control, which is no cover crop, were actually, so these three cover crops were planted on soils of different test levels, 10, 20, and 30 milligram per kilogram, also called ppm, Olsen P. That's the standard within our region. Experimental design was RCBD. The crops were grown using field condition parameters, irrigated at 3 days intervals. So whenever there was no rain, it was a holiday for this young scientist. Again, in winter, what we did was to cover the cover crops to prevent rain and snow from dragging the nutrients into surface waters. These are the nutrients that I am supposed to go and collect. And therefore, after winter, that is in spring, we set up this bench as 5% slope. And what we did was to put the snow on the cover crops. So I'm sorry you can't see the cover crops here. But as you can see, there's this light that you see over there. The light was erected above the setup at a reasonable height to ensure vertical distribution of heat, just like what happens on the field. We tried, but we couldn't get runoff. So the soil thawed and the leachates were collected in the pails you see under the bench here. So these white plastic bottles couldn't collect the runoff we wanted. The amount of snow that we applied were 25 centimeters for the first scenario, and then we repeated again with 10 centimeters of snow. With the rainfall, a simulator height 3 meters above the box was erected at an intensity of 50 millimeters per hour, and 40 millimeters of runoff was collected. Results indicated an increase of dissolved reactive phosphorus with soil test phosphorus for the Snowmelt Leachate, which tells us that soil test phosphorus is a problem. Again, we found with the cover crops themselves that there was no difference between all the cover crops and the control. However, for the cover crops themselves, there were differences where annual ryegrass gave out more DRP than the other cover crops, oat and red clover. Going back to the rainfall runoff, we again found out that soil test phosphorus, as they increase, there's also an increase in dissolved reactive phosphorus. And therefore, this gave us the sense that soil test phosphorus should be paid attention to. And therefore, low soil test phosphorus may be better than high in terms of managing phosphorus losses. Going back to the rainfall again, that is with the cover crops themselves. This time, for the cover crops, there were no differences between them. However, between control and then the oat gave out more phosphorus than the control. Note, with the snowmelt, it was ryegrass, which was a grass. With this one, which is rainfall, it is the oat, which is also a grass. What is it with the grasses? My lab analysis of the biomasses research that I've been doing also aside, have found out that when you freeze-thaw these cover crops, the same amount of cover crops, and extract nutrients from them, you get equal amounts from them, equal concentrations. But because the grasses usually have higher biomasses, if you multiply their biomass by the concentration, they give out more in terms of this kilogram per hectare. And that is why probably in this one, we are seeing the grasses giving out more dissolved reactive phosphorus in the scenarios that we set. To conclude it all, I will say cover crops impact dissolved reactive phosphorus amount differently depending on the medium of runoff, which is either snowmelt or rainfall. And therefore, species selection is very essential in this research, where clover may seem promising in this research. Also, soil test phosphorus is critical in phosphorus management because the DRP losses we found out increased as soil test phosphorus also increased. As our take home, I would say red clover and low STP, which is the soil test phosphorus, may be our savior. Few references here And I will say thank you all for your time. It has been wonderful. Happy being with you. Thank you so much.